Hello everyone. In this session we have to discuss part 3 of chapter 8, and this will include the content that is endomembrane system found in a cell. That is, we have to see detailed structure of endoplasmic reticulum and its function. Then we will proceed to structure of Golgi bodies, structure and function. And then we have to see lysosome, its types and function. This is part 3 of chapter 8, that is cell, the unit of life. We will concentrate in this part the endomembrane system, endoplasmic reticulum, then we'll go for Golgi bodies, and in the last of this part we will see lysosomes, types of lysosome. You know, children, endomembrane system, what we mean by? You know, in a cell there are different type of cell organelles are there. That is, some of the cell organelles, these are not related to other, but a few cell organelles, these are related, these are having integrated structures. So such, that is, organelles, they make endomembrane system. Each of the membranous organelles is distinct. You can see that this is chloroplast, right? Or let this one is, that is mitochondria. These are distinct. These are not connected to. This is that is chondria, this is mitochondria. But you see, many of the organelles are there which are considered together as endomembrane system. Why? Because their functions are coordinated. They are, that is, related to each other. Endomembrane system that includes endoplasmic reticulum, in short we will call it as ER, Golgi bodies, lysosome and vacuoles. The functions of all these, that is integrated, and these are coordinated together. That's why they will constitute endomembrane system. Is it very much clear? Chloroplast, mitochondria, centriole, all these are not related to each other. Therefore, they don't make, that is, endomembrane system. Since function of the mitochondria, chloroplast, peroxisome are not, that is, coordinated, as I told you, that's why we will not call them as component of, that is, endomembrane system. These are not considered part of endomembrane system. The first, that is, component of the endomembrane system we have here, that is endoplasmic reticulum, made up of two words, endoplasm and reticulum. Endoplasm means what? That is, if this is a cell, this is nucleus. That is, roughly we can divide the cytoplasm of this in two parts, that is outer layer and the inner part. Endo means the cytoplasm which is surrounding the, that is, nucleus, that is endoplasm. Reticulum means a net-like structure. The name endoplasmic reticulum was given. A network or reticulum of tiny tubular structures which are scattered in cytoplasm is known as endoplasmic reticulum, ER. And ER, you see, that is, if this is a cell, let this is the endoplasmic reticulum, a tubular structure. So it has divided the cell into two parts. ER divide the intercellular space into two distinct compartments, which is inside the endoplasmic reticulum, known as, that is, luminal, and which is outside will be known as, that is, extraluminal one. The compartments are because of endoplasmic reticulum, important one. Endoplasmic reticulum, it gives compartments in cell, right? Now you see that this is the nucleus. I told you that this is the endoplasm. Outside that will be, that is, we can call that is ectoplasm. Inside the endoplasm, close to the nucleus, these are the tubular structures are found, and that's why known as, that is, endoplasmic reticulum. If I'll make that this is the nucleus here, that is attached to the nuclear membrane, these are the, that is, tubular structures like this. We may call that this is the endoplasmic reticulum, like this. These are found. You see, endoplasmic reticulum, that is, three type of, that is, structures are found. One is known as cisternae. These are tubular, which lies parallel to each other, as you are seeing here. These are the cisternae, and they have on the surface, that is, they have, that is, ribosomes are found attached. Ribosomes, eukaryotic cell, the ribosome will be a TS type of ribosome. That is, sometimes the endoplasmic reticulum is in the form of vesicle, and sometimes it is in the form of, that is, branched tube. So this endoplasmic reticulum, that exists in three forms. One is, that is, cisternae, that is, parallel tubes as I made here. that in the form of vesicles, spherical structure, or in the form of that is branched tubules. We have to see detail of this. Component of endoplasmic reticulum. Yes, number one is cisternae, long, flat, parallel, sac, interconnected structures. These are found in the cells which are actively engaged in protein synthesis. Why? Because on these are the interconnected parallel tubules like this, because we have the ribosome, and you know ribosome is the site of protein synthesis. That's why the cisternae are mostly found in cell which are engaged in that is protein synthesis. Is it right? Tubules, these are that is branched or unbranched structure, that is fine tubular structures. They may be branched or unbranched one, forming a reticular along the cisternae or vesicles. They are free of ribosomes, and these are involved in the cell which are involved in lipid and sterol synthesis because they do not have ribosome on surface. Vesicles, these are oval membrane bounded structure. These are also free of ribosome spherical structures, they told in that diagram also. Structure of endoplasmic reticulum is similar to that of Golgi bodies. However, what happens? In endoplasmic reticulum, cisternae, vesicles, tubules are isolated in cytoplasm. They do not form a complex. You see, the same thing, cisternae, tubules and vesicles are found in Golgi bodies, but here in case of Golgi bodies, all these are making a compact structure near to the nucleus, while in case of endoplasmic reticulum, these are found scattered throughout the cytoplasm in isolated form. Golgi bodies, as I told you, this is a localized one, while the endoplasmic reticulum is found widespread in cytoplasm. Is it? Endoplasm reticulum is often termed as systems of membranes. This is called systems of membrane. Okay. Come to the next now. Endoplasmic reticulum bearing ribosome on the surface is called, that is rough endoplasmic reticulum, as I told you. And if the ribosomes are not associated with ER, known as smooth, that is endoplasmic reticulum. We can differentiate, that is rough endoplasmic reticulum. You can recall that I had made that this is cisternae is there. These are the system of parallel tube like this which are interconnected, and the ribosomes, these are attached to this. So ATS ribosome bind to the surface larger unit. This will be larger unit with the help of two glycoprotein, that is ribofurans first and second. Is it? And now that is, this is more stable structure, mainly composed of cisternae. As I told you, these are abundantly found in cells actively engaged in protein synthesis. For example, the cells of liver and pancreas. A smooth endoplasmic reticulum, these are that is tubular structure, or these are vesicles. They do not have ribosome, therefore the surface is smooth, less stable, mainly composed of tubules or vesicles, abundantly found. Yes, where? The cell which are concerned with glycogen and lipid metabolism. In a case of animal cell, lipids such as steroid hormones and synthesis in SER, adipose tissue, interstitial cells, muscles of glycogen, storing liver cells, and adrenal cortex. All these are engaged, that is in steroid synthesis, lipid synthesis, or glycogen storage. They have that is smooth endoplasmic reticulum. It is called agranular. This is called that is granular one. Is it clear? Now, functions of SER, that is we can correlate easily. Smooth endoplasmic reticulum, fat and steroid hormone synthesis. These are found in that is gonads. That is because here steroids hormones are glycogen metabolism, detoxification of drugs, is it? And that is muscle contraction by release and uptake of calcium ions, formation of organelles such as spherosomes and glyoxysomes, and cytochrome, that is P450 in endoplasmic reticulum. It acts as enzyme which functions detoxification of drug and other toxic substances. You can remember it. On other side, functions of the rough ER, can you recall? It provide site for protein synthesis, formation of nuclear envelope and plasma membrane, smooth ER is also formed from, that is rough ER, and synthesis of the lysosomal enzyme. You know lysosome, these are the bag-like structure filled with different type of enzyme. These are protein in nature. Protein is synthesized where? In ribosome, and the ribosomes, you know, these are found attached to, that is endoplasmic reticulum. Which one? Rough or smooth? That is rough endoplasmic reticulum. Is it right? Come to the next now. We have to say Golgi apparatus. Yes, that is Camillo Golgi in 1898 first observed densely stent reticular structure near to the nucleus. If this is a plant, that is animal cell we have here, and this one is the nucleus, he found close to the nucleus, that is a densely stent area, and where a compact structure was seen. That structure was named after his name, that is Golgi bodies. As I told you while discussing ER, that is Golgi body also consists of cisternae, tubules and vesicles. But the differences are there. What are cisternae? You can see in diagram, that is here close to the nucleus. These are many flat disc-shaped cisternae, that is of 0.5 to, that is 1 micrometer in diameter. These are stacked parallel to each other. I can make it that these are stacked like this, that is parallel to each other like this. These are placed one above the other in group. Varied number of the cisternae are present in Golgi complex. That is, these cisternae are concentrically arranged near the nucleus with distinct convex or cis or forming face and concave, trans or mature face. The cis, this face is close to nucleus. This is convex, this is cis, or that is known as, that is forming face, while towards the plasma membrane the face is concave, that is concave one, or known as trans phase or known as maturing phase. You have to remember near to the nucleus that these are here, that is near to the plasma membrane, towards the plasma membrane the surface is known as, that is trans, or known as that is mature surface, or this one is, that is the concave one. Important point this is. Tubules, these are found, that is attached to this. These are the branched structures, and at the mature surface, that is these vesicles are being cut off. Tubules are small, flat, interconnecting structure arising from periphery of the cisternae due to, that is fenestration. Vesicles, these are large, rounded sac-like structure found at the edge of cisternae, and these are pinched off from the tubules. The tubules, they will cut off these, that is vesicles. Sometimes Golgian vesicles, Golgian vacuoles are found. These are the large, comparatively larger spherical vacuoles produced as maturing surface here. These are the large one, and actually these are filled with some granular amorphous substances, and some of these, they will help in formation of, that is lysosome. Right? You can see the diagram of this Golgi body. That is, I can say this is a trans phase. This is what? That is newly formed vesicles. These are, this is the cisternae, this is cis phase, that is convex phase, and this will be the, okay, that is, I told you, that is cis phase. Yes, this is cis phase. This is also known as, that is forming phase, right? Or that is known as, it is called convex phase. It is towards nucleus, while towards plasma membrane the phase is, that is the concave one, or also called as, that is trans phase, or also that is called the mature phase. So here this is the Golgi apparatus. Is it? This is, that lumen is there in between. This is the secretory vesicles are there. Trans phase, that is mature phase, this newly formed vesicles are there, and definitely the nucleus will be towards this side. This is nucleus here, this is nucleus. Close to nucleus this Golgi body is present. Now functions of the Golgi body we have to discuss. Golgi apparatus, that is important. Its principal function is, that is packing material to be delivered either to intracellular or, that is secretory outside the cell. What will happen? A very interesting diagram I can give you here. Let this is the plasma membrane. Okay, this is, or let this is that is endoplasmic reticulum. Here these are the ribosomes. Ribosome, they will make the protein, ribosome. That is the endoplasmic reticulum, which is SER. What is this? This is smooth endoplasmic reticulum. This one is, that is rough endoplasmic reticulum. That is, here the protein will be synthesized, the protein will be pinched off here, and this protein will be a part of, that is, Golgi body. Now that will go into Golgi bodies, right? And here in Golgi bodies, that is this substance produced here in ribosome, transported by SER, here in Golgi body, that will be modified. It will get, that is, unite with some other substances, and ultimately that will be pinched off in the form of vesicles. Either these vesicles will be used inside the cell, or they will be secreted outside the cell. So this is the main function of Golgi body, that is, packing will be there. The substance synthesized at different part of the cell will be brought to the Golgi bodies. Either this is the carbohydrate are synthesized here, or the fats are synthesized here. They will also be brought to the Golgi bodies, and the packing of different, that is, substance will take place here. Okay, now we can read it out. Packing material, that is either used inside the cell, intracellular, or that is secreted outside the cell. Material to be packed in the form of vesicles, either it comes, that is, from ER, endoplasmic reticulum, with the cis phase of the, this is cis phase. What is this? This is nucleus. This is cis phase. These substances will be added towards cis phase. Okay, and they will start to move towards the maturing phase, where they will be released. Therefore, Golgi body apparatus, it remain in close association with endoplasmic reticulum. Number of the proteins synthesized by ribosomes, you see here, that is on endoplasmic reticulum, are modified in cisternae of Golgi body before they are released from its trans phase. Is it very much clear? Golgi apparatus is important site of formation of glycoprotein. That is, when the carbohydrate will combine with protein, will be known as glycoprotein. The process is known as glycosylation, important. Or that is glycosidation, when the carbohydrate, when it combines with, that is, lipid, that will be known as glycolipids. The process is known as glycosidation. Glycosylation, and that one is, that is, glycosidation. Acrosome of the sperm is modified, Golgi body. The tip of this, that is, here this is the large nucleus, that is acrosome will be, this is modified Golgi apparatus. Root cap cells are rich in, that is, in root, this is the root cap. The root cap cells, they have, that is, much more Golgi bodies which secrete the mucilase for lubrication of the root tip, and Golgi apparatus, it helps in recycling of the broken plasma membrane during endocytosis. Means, that is, plasma membrane, Golgi bodies, and ER, all these are interconnected. Is it right? Any doubt? Can we proceed to the next now? The main function of the Golgi body is packing material. The protein will come through endoplasmic reticulum. Carbohydrate and fat will also come. All these will be added to cis phase, and then it will be modified, and then will be released at trans phase in the form of, that is, vesicles. Okay. Come to the next, the last part of this, that is, session. Lysosomes, also known as, that is, suicidal bags. Discovered by Christian Didieu. He is credited for the discovery of this. They are single membrane bounded small vesicular structure rich in hydrolytic enzyme. You see, children, this is a bag-like structure. I told you that was the Golgi body. That is from where it will be formed. That is Golgi body or Golgi apparatus. What is this? Is the trans phase of this. Trans phase. That is, this is mature phase. Is it mature phase? This one is. That is from this Golgi body. The lysosome will be cut off. And it is having, that is, enzyme, hydrolytic enzyme. It is having enzyme which can digest almost all the things. It is surrounded by, that is, single unit membrane. This is unit membrane. This is membrane we have here in lysosome. single unit membrane. It contains about 50 hydrolytic enzyme. 50 hydrolytic enzyme inside the lysosome. They can digest the carbohydrate. They can digest all type of protein. They can digest all type of fats. They can digest DNA. They can digest RNA. Almost every type of chemical found in a living cell can be digested by this. Okay, that is these 50, very important point, 50 hydrolytic enzyme, acid hydrolases, which can digest most of the biological substance. Acidic condition is maintained by pumping proton into the interior. Hydrogen ions, the proton, will be pumped here inside the lysosome so that the pH inside, that will be low, acidic conditions will be there. That's why known as, that is acid hydrolases. These are important. That the point here we have, these organelles show polymorphism. You have to see what we mean by polymorphism. In next slide I'll tell you. With the exception of mammalian RBC, they are reported almost all cells. That is, very frequently these are found in case of, that is, animal cell. Is it? In plant cell, the large central vacuole function is lysosome. So in higher plant, lysosome are less frequent, but number of the lysosome, that is high in case of fungi. You have to remember. Periplasmic space, even in bacteria, that is in between the cell wall and plasma membrane. If this is a bacterial cell, this is plasma membrane and this one is cell wall. This is plasma membrane and this one is cell wall. The space in between two is known as periplasmic space. Now come, that is type of lysosome. Before, that is, we discussed, you can see a beautiful diagram of this. That is lysosome. 4 type of lysosome. This one is that is Golgi body, right? That is, as I told you, from Golgi bodies, the lysosome will be pinched off. This is the lysosome. This is lysosome in the form of cycles are cut off from. It is known as primary lysosome. Okay, primary lysosome, as it is having different type of enzyme, that this is any food is taken by endocytosis by cell. This food will combine with the lysosome and now that will become the secondary lysosome and it will able to digest all the content of the food here. Digestive are also known as, that is, it is called secondary lysosome. The digested food that will diffuse out inside the cell, and the undigested food which is left here, that is known as residual lysosome or residual body. So this is number 1, primary lysosome. This is second, number 2, secondary lysosome. This is the third one, undigested part of the food is there inside and that will be thrown outside, that is exocytosis. Exocytosis will be there. Okay. Now one more type we have, at the time of need, when that is starvation is there or the energy is required, the cell is not getting that is supply of food, the lysosome, this lysosome, it will select the least, that is important component of the cell, that this is old mitochondria. Lysosome will take this old mitochondria and the content of the old mitochondria will be digested here. Here in this case, it is known as autophagic vacuole. If the need is there, that is based on priority, lysosome will like to select the different cytoplasmic organelles and will digest. The digested food will diffuse inside the cell so that the cell will survive. Therefore, it is known as, why? Because it has taken its own component and killed and digested. That's why these are known as suicidal bag. Is it right? Suicidal bag. So 4 type of, this is known as autophagic vacuole. 4 type of lysosome. Therefore, lysosome is called, that is shows different forms. It shows polymorphism. It is called polymorphism. Okay. Now we can read it out. Lysosome, there is no need to go in detail now. Primary lysosome, small vesicles, newly formed structure produced from Golgi bodies at transphase definitely, and it contains inactive enzyme. Secondary lysosome, also called Heterophagosome, digestive vacuole, that is secondary lysosome. How that will be formed? They will be formed when phagosome, that is food, fuses with already existing lysosome. They contains enzyme. The enzyme will digest the food. The digested food will diffuse out into the cell, and that remaining part is left, which is known as, that is, residual body. Autophagic vacuoles, these are formed by the union of many, that is primary lysosome surrounded with the vacuolar membrane, or that is old, that is cytoplasmic organelle, and they will digest, will get the energy, known as autolysis or autodigestion. Therefore, we can say in the last, functions of the lysosome: intracellular or extracellular digestion and autolysis caused by lysosomal enzyme. That is, enzyme name is, that is cathepsin, is play important role in disappearance of tail metamorphosis. You know, in tadpole, that is, the tail is there. During metamorphosis, the tail will disappear. That is known as metamorphosis. This disappearance of the tail is, that is, by lysosomal enzymes. Is it right? Okay, that is part 3 of chapter 8. Thank you.